आप बोलो सर।
Good morning. Sound कम कर दीजिए. Friends, today we will discuss the drugs acting on parasympathetic system are cholinergic agonist, not antagonist. Broadly, we can classify nervous system into broad area. One is the central nervous system and other is the peripheral nervous system. This central nervous system itself further classified. One is the brain, another is the spinal cord. And this peripheral nervous system, the neurons, nervous system get broadly classified into two broad areas, motor neurons and sensory neurons. Sensory neurons, which carry impulse from periphery to the CNS, or you can say from the sensory organ to the central nervous system. When you discuss about the motor neurons, which carry masses, or you can say which carry impulse from CNS to other organs, that is the effective organs, you can say. And these motor neurons, broadly two types. One is the somatic nervous system, one is the autonomic nervous system. The somatic neurons nervous system, which regulated by your will, means the neurons, those neurons, broadly those carry impulse from higher center to skeletal muscles that get controlled by your will. Autonomic nervous system is not controlled by your will. And when we are discussing about the autonomic nervous system, it further classified sympathetic and parasympathetic. And my dear friends, this autonomic nervous system, I can say, is a base of pharmacology. If you know autonomic nervous system, if your concept is very clear in autonomic nervous system, you will be enjoying for study of rest of the other pharmacology chapters. I will try to much focus, and whatever problem is there, you don't hesitate to write a mail. That is h@yadav@gmail.com. This is the somatic nervous system. The neurons originated from the heart center, directly innervated to the organ, effective organs. And the neurotransmitter is the acetylcholine. That is the totally controlled by your will. And the receptor is the NM type. We will discuss later on. There is no any ganglia in somatic nervous system. If you see the autonomic nervous system, that get further classified into parasympathetic and sympathetic nervous system. Both are having ganglia. And the ganglia, in the case of the parasympathetic nervous system, these ganglia are present very close to the organs. And that's why this length of this post-ganglionic neuron fibers is comparatively smaller than the length of preganglionic fibers. And here the neurotransmitter is acetylcholine in both cases, in preganglionic fibers as well as postganglionic fibers. When you are discussing about the sympathetic nervous system, friends, when you dissect out any mammals, rat, rabbit, mice, you will see there is a group of the ganglions very close to and parallel to the vertebral columns, and that's why it's known as the paravertebral ganglia, because these ganglia, a chain of the ganglia present parallel to the vertebral columns, and because these ganglia are very close to the vertebral column, that's why the length of these preganglionic fibers is smaller as comparison to the postganglionic fibers. In the case of the parasympathetic nervous system, acetylcholine is the neurotransmitter in both sides. But in the case of sympathetic nervous system, acetylcholine is the neurotransmitter in preganglionic fibers, and in postganglionic fibers the neurotransmitter is the norepinephrine. Here, and except few organs, sweat gland, blood vessels, hair follicles and adrenal medulla, where these organs are innervated by sympathetic nervous system, and neurotransmitter is acetylcholine. That are the exception, and we can block the activity by giving the acetylcholine antagonist, that is the atropine. Here the neurons originated from the sympathetic nervous systems directly innervate to adrenal medulla, and these are the receptor, and further activate the epinephrine and norepinephrine, this ratio 80:20. Here is acetylcholine release activate the glands. These are the exceptions. When you are discussing the parasympathetic nervous system, or you can say the cholinergic agonist, and before the discussing of the cholinergic agonist, we can differentiate broadly what is the effect of parasympathetic system and what is the effect of the sympathetic system. If you see the organs, individual organs, the effect of both symptoms is almost completely opposite to each other. For example, on the eyes, in parasympathetic nervous system, receptor or acetylcholine present on the circular muscles, constrict the circular muscles, constrict the pupils, that is called meiosis. Salivary gland, increase the salivation. Heart rate, decrease the heart rate, decrease the force of contraction. Lungs, constrict the bronchi. Stomach. Stimulate the stomach means increase the peristaltic movement. We are discussing about the parasympathetic system. Liver. Stimulate the release of bile. And intestine similarly increase the peristaltic movement. Bladder, constrict the bladder. And we are discussing about the sympathetic system. Here people get constricted, meiosis is there. Here people get dilated. Its receptor predominantly present on the cervical muscles, constrict the pupil. These receptors present on the radial muscles. When radial muscles constricted, you see that dilate the pupils. In broad way, the effect on the eye is opposite in the case of the sympathetic nervous system. Salivary gland, stimulate the salivation, inhibit the salivation. Heart, decrease the heart rate, increase the heart rate. Lungs, constrict the bronchi, dilate the bronchi, especially by beta 2. Stomach, you see, stimulate the digestion, inhibit the digestion. It increase the peristalsis, decrease the peristalsis. Liver, stimulate the glucose release. Kidney, similarly, stimulate the epinephrine and norepinephrine release. And in intestine, it increase the peristaltic, it decrease the peristaltic. Bladder, it constrict the bladder, it relax the bladder. If you see the effect of any organs, effect of parasympathetic system is totally opposed by the sympathetic system. Effect of the sympathetic system is totally opposed by the, and there is a control of both of them in this way. They are controlling. Here the ganglia are very close to the vertebral column, that's why known as the paravertebral ganglia. Here the ganglia are present very close to the organs. When you are discussing about the receptor, the cholinergic receptor are broadly classified into two types. One is the nicotinic receptor and one is the muscarinic receptor. Nicotinic receptor is ligand gated, means once the receptor get activated, it directly open the ion channels, suddenly open the ion channels, and effect is very fast. And this nicotinic receptors are further get classified into NN present on the basically peripheral neurons. In the peripheral neurons, this NN receptor present into the autonomic ganglia. NM receptor present on the skeletal muscles. Action is comparatively fast. When the receptor get activated, it suddenly open these ion channels. When we are discussing about the muscarinic receptor further get subclassified into 5. That is M1, M2, M3, M4 and M5. These nicotinic receptors are basically ligand gated ion channels. This muscarinic receptor are G protein coupled receptor. Receptor act on the drug act on the some G receptor, some secondary messenger get generated, secondary messenger will generated it, and it phosphorylates some protein. When it phosphorylates some protein, it opens the channels. Means the process is something is the lengthy. It is the time consuming process as compared to this ligand gated ion channel. It is very fast. And M1, M2, M5 is the belonging to the GQ, and M2 we will discuss later on what is the GQ and what is the GI, and these are the GI. And we are discussing about the G protein coupled receptor. Is a family of GTP binding receptor. The G protein coupled receptor of 7 alpha helical chains present on the cell membrane. On the above the cell membrane is the receptor binding site. In the cytoplasm, it is the site for the binding of the G protein. It means GDP. At the resting is okay. This G protein further classified into alpha, beta, and gamma subunit. Alpha subunit fall into 3 families. GS means stimulate the adenyl cyclase, increase the cyclic AMP. Adenyl cyclase act on the AMP and produces cyclic AMP. GI inhibit the adenyl cyclase, decrease the formation of the cyclic AMP. Cyclic AMP is the active form of the AMP. It is the secondary messenger. Okay, GQ, where M1, M3 and M5 are belongings, activate the phospholipase C, increase the production of the IP3 and DAG. Before going into the further receptor, first thing is the production of the agonist, endogenous agonist, and that is the acetylcholine. How acetylcholine is synthesized into the neurons, and how this synthesized neuron get released. This typical end plate of the neurons. Here the choline get transported into the exoplasm through choline sodium cotransporter. Once this choline enter into the exoplasm, or you can say cytoplasm of the neurons, this choline get converted into the acetylcholine, and the enzyme is responsible for the choline acetyl transferase, which facilitate the production of this acetylcholine by the choline and acetyl coenzyme, and with the help of ATP. 3 molecules combine and produces acetylcholine. Once acetylcholine get generated into the cytoplasm, until or unless it get protected by some vesicle, it get degraded. And for the prevention of its degradation, its metabolization, this formed acetylcholine get actively transported into the vesicle, where it get protected from its metabolism, and it's metabolized by acetylcholine esterase, which degrade acetylcholine. And once this acetylcholine get actively uptake into the vesicles, it get totally protected and stored. You can say this acetylcholine get stored into the vesicles, and during the nerve stimulation, or you can say the membrane potential is sufficient to increase the sufficient calcium level into the cytoplasm. You can say this vesicle get fused into the membrane of these neurons, and once vesicle get fused, acetylcholine is able to release into the synapse. It may be synapse, this may be neurons, and you can say where it is synapse. When it get directly innervated to the end organs, you can say it is the effective organs. It may act on the effective organs, receptor of the effective organs. Otherwise, this acetylcholine get further degraded by acetylcholine esterase into the choline and acetate. And this choline, it further reuptake and started the synthesis of acetylcholine. And it is the feedback regulation of which can inhibit this vesicle release fusion. When the acetylcholine into the synapse is sufficiently high, it acts on this presynaptic neurons and further regulate the release of these vesicles. And whatever the choline, acetylcholine get released, it may act on the effective organs and may exert their action. And when it get degraded, it degraded by the enzyme acetylcholine esterase into the choline and acetate, and choline get further removed. And this is the whole cycle which continuously going on. This acetylcholine here act on the receptor and exert action. The action depend upon the type of the receptor. When it is the muscarinic receptor, act on the muscarinic receptor. Muscarinic receptor are the G protein coupled receptor. Here in the inactive state, this G protein, it that is the consist of the 3 unit alpha, beta, gamma. Once receptor get active, once agonist bind on the receptor, and once there is some conformational change get happening into the receptor, and these alpha molecules earlier get bind by GDP, it get replaced by GTP, and once this alpha molecules carrying GTP, it activate phospholipase C, and when phospholipase C get activated, it act on PIP to produce IP3 and DAG. And this PIP, phosphoinositol diphosphate, get converted into IP3 inositol triphosphate. It is the active form of second messenger. This IP3 activate the calcium channel here, and calcium channel get open. When calcium channel get open, calcium enter into the cell. When calcium into the cell, it produces action by the calcium carmodulin by through different enzyme, and one enzyme is the protein kinase, and produces protein kinase get phosphorylated and response get produces. And this PIP get converted into IP3 once, and that one thing is the DAG, diacylglycerol. This DAG, how DAG exert actions? This DAG directly activate the protein kinase, and this protein kinase phosphorylate the other proteins by the help of ATP. And once protein get phosphorylated, it's the activate some directly able to produce exert actions. It means the whole this is the whole story how it is happening. And when you get both, this previous one is the receptor present on the muscarinic receptor, this receptor on the ligand gated receptor. Suddenly effect get exerted, that is ligand gated and channels present on the NNNM here. When receptor agonist bind, directly open the calcium and directly open the channels. When the directly open the channels, calcium level get increases and produces action. It's the quick response as compared to the previous one. When you are discussing about the muscarinic receptor may be NM, and these are the effect is the excitatory, contract the skeletal muscles because this NM present on the skeletal muscles. Increase the cations because this N and M are the ligand gated. Open the these channels and produces contraction of the skeletal muscles. Agonist is acetylcholine, succinylcholine, nicotine. These are the antagonists. D-tuberculin is the mode, pancuronibromide and so on and so forth. These are the neuronal type. Receptor present on the autonomic ganglia or adrenal medulla, and when it present on the autonomic ganglia, it further responsible for the firing of the postsynaptic neurons, and in the adrenal medulla it responsible for the release of epinephrine and norepinephrine. These are the agonists and these are the antagonists. Now we see the muscarinic receptor, broadly classified into 5 types: M1, M2, M3, M4 and M5. M1, M2 and M3 are well elucidated. This M1 present on the GIT muscles and brain. M2 present more abundantly on the cardiac muscles and brain. M3 receptors are more abundantly present on the adipose tissue and brain. You see, there is one common thing. All 3 receptors, M1, M2 and M3 receptors are present on the brain. And a specific location of M1 receptor on the GIT, M2 receptor on the heart, especially SA node, AB node, and M3 receptor on this brains and not smooth muscles, not at both sides. That is the smooth muscles. Sorry, M3 receptor present on the smooth muscles and brain. Functions: gastric acid secretion, CNS excitation. Agonist: oxytimorin, antagonist: parangepine, terangepine. M2 receptor decrease the heart rate. We will discuss later on how decrease the heart rate, decrease the conduction velocity, decrease the contractility, and in opposite to the M1 receptor with the CNS excitation, the CNS inhibitory action is there. And agonist is the methacholine and antagonist is the galantamine and so on and so forth. These are the M3 receptor. Increase the secretion, gastric secretion, increase the contractility, increase the smooth muscles and CNS excitation. It basically present on the stomach. These present on the other smooth muscles. M3, and these are the agonists and these are the antagonists. And M4 and M5 are not well elucidated. When you are discussing about the cholinergic drug, cholinergic agonist, or you can say broad way cholinergic drugs, further classified into 2 types. One is the cholinergic agonist, directly act on the cholinergic receptor, and it is the acetylcholine esterase, indirectly activator of the cholinergic receptor by inhibiting those enzymes which degrade the acetylcholine, or you can say it is the acetylcholine esterase inhibitor. And when we are discussing about the cholinergic agonist, it further classified into 2 types. One is the cholinergic esterase, another is the alkaloid. Choline esterase, acetylcholine, methacholine, carbacholine, and bethanoicol. And alkaloids, they are natural compounds, plant derivatable substances, alkali in nature. It may be muscarine, pilocarpine, aerocholine, pilocarpine is commonly used. And these are the indirectly acting cholinergic drugs, inhibit the acetylcholinesterase. We will discuss later on. It further classified into reversible acetylcholinesterase inhibitor, irreversible acetylcholinesterase inhibitor, and reversible further classified into carbamide, 5-stupine, neostupine, pyridostigmine, so on and so forth. And the acridine, tacrine, and irreversible is the organophosphate. That is a very common responsible for the common poisoning, and the carbamides. We will discuss this portion. We will discuss later on. Today we will discuss this cholinergic agonist only. Okay, how the cholinergic agonist able to exert their action? We will discuss acetylcholine as a prototype. When you are discussing about the heart, it acts on the M2 receptor on the SA node and hyperpolarized by opening the potassium channel, and inhibit the phase 4 diastolic depolarization. When it inhibits the phase 4 diastolic depolarization, it means it increases the time period of phase 4 to achieve the threshold value for the rapid firing. And when it increases the time period of phase 4, it means heart rate get decreases. Heart rate get decreases, conduction velocity get decreases. You can say when you give the high dose, totally AB block is there, and refractive period get increases, and PR interval increases, and when you give the higher dose, it may block the arterioventricular conduction, and force of the arterial contraction is decreases, force of the ventricular contraction get also little bit decreases. It total net effect of acetylcholine on the heart is decrease the heart rate, decrease the heart rate, decrease the force of contraction, and decrease the rate of impulse generation and impulse conduction. Negative chronotropic, negative dromotropic, negative inotropic. And effect on the blood vessels. It acts on the endothelium, increase the production of nitric oxide by the calcium calmodulin process, and this nitric oxide generated by the endothelium rapidly migrated to the surrounding smooth muscles, and smooth muscles get dilated, relax, and you can say the action of this acetylcholine in a healthy blood vessels where the endothelium is not ruptured. When the endothelial dysfunction is not there, it dilates the blood vessels. And when you are discussing about the penis, it dilates the cavernosal vessels, corpus cavernosal vessels, and may allow to the filling of the blood that is responsible for their action. Friends, when you are discussing about the smooth muscles, most of the smooth muscles get constricted through M3 receptor. GIT tone and peristalsis get increased, sphincter get relaxed. When the peristaltic get increased, sphincter get released, it means complete evacuation of the bowel may be there. Abdominal cramp may be there. When you are discussing about the bronchial muscles, also by the M3 receptor, it constricts the bronchial muscles. and asthmatic peoples are more sensitive toward the bronchospasm, dyspnea, and there is an attack of the asthma is maybe there. When you are discussing about the action of the acetylcholine on the glands, most of the glands are innervated by M3 receptor. It increases the glandular secretion, and some by M2 receptors. Sweat glands, when activated, sweating is there. Salivary gland when activated, salivation is there. Lacrimation is there, and increases the tachybronchial secretion also. And eye, it act on the we had already discussed on the previous slides. It because cholinergic receptor present on the smooth muscles, it constricts the smooth muscles, and pupil side get decreases, and you can say meiosis is there. When you are discussing about the nicotinic action, previous one is the muscarinic actions, and now see the nicotinic receptor. Nicotinic receptor get further classified into two types, NN and NM. NM present on the skeletal muscles, predominantly present on the skeletal muscles in plate. NN present on the peripheral neurons, especially into the autonomic ganglia. When you give acetylcholine as such, it acts on the heart, decrease the heart rate, dilate the blood vessels. It means when the acetylcholine acts on the heart, decrease the heart rate, dilate the blood vessels, the overall effect on the blood pressure of acetylcholine on the blood vessel, blood pressure is decreases. And there is a very interesting things, my dear friend, you all may note it. It is a very interesting things. When you give acetylcholine after giving the atropine, when you give acetylcholine after giving the atropine, what is happening? Previously, when you give the acetylcholine, it decreases the heart rate, it dilates the blood vessels, BP get decreased, bradycardia was there. When you give acetylcholine in those subjects that get already administered by atropine, atropine block the acetylcholine effect on the heart, and you see it means there is no effect of acetylcholine that get observed on the heart. It means acetylcholine, effect of acetylcholine on the heart is prevented by the atropine, because atropine is an antagonist of acetylcholine, bind on the same receptor, inhibit the effect of acetylcholine. But acetylcholine act on the ganglia, penetrate into the ganglia, activate the ganglionic receptors, and increase the firing of both neurotransmitters. In the case of sympathetic neurotransmitter, it increases the firing of norepinephrine, and there is an unopposed action of norepinephrine on heart, because cholinergic effect gets blocked by giving the atropine. And when it acts on the parasympathetic neurons on the ganglia, it facilitates the release of norepinephrine, and there is an unopposed action of the norepinephrine on the heart, increase the heart rate, tachycardia is there. It means Effect of acetylcholine as such is bradycardia. When you give acetylcholine, after the giving the atropine, tachycardia may be noted due to the ganglionic action. Skeletal muscles, by inotropopheric by some stimulation on the electric stimulation on the dermal patches, it may causes the contraction of the skeletal muscle fibers. And when you give the intra-arterial in high doses, it may causes twitching, muscular twitching and fasciculation. CNS overall. When acetylcholine injected, IV does not penetrate the blood-brain barrier. When you give directly into the brain, it may produce the arousal response followed by the depression. Cholinergic drug, which can cross the blood-brain barrier, may exert the complex behavior. Something is excitation, by something is a depression, because all the receptor M1, M2 and M3 are present on the brain, and therefore effect is the complex. Now comes on the interaction of acetylcholine. Acetylcholine is metabolized by acetylcholine esterase, and when you give the acetylcholine esterase inhibitor, metabolism of acetylcholine is get impaired and the cholinergic effect get dominated, because acetylcholine does not metabolize. Methacholine and carbachol, methanicol may potentiate action. Adrenaline is the physiological antagonist of acetylcholine, and atropine is the antagonist of acetylcholine. Uses rarely used. Methacholine earlier used for the prevention of the proximal supraventricular tachycardia, but not nowadays. There are other safe drugs are available. Methanicol has been used on the post-operative and non-obstructive urinary retention, and side effect is the bleeding, involuntary urination, sweating, falling BP, and bronchospasm, especially into the asthmatic peoples. And these are the alkaloids. Pilocarpine is the more commonly used alkaloids obtained from the Pilocarpus microphyllus, another species having the prominent muscarinic action, and also stimulate the ganglia by the ganglionic muscarinic receptor. Pilocarpine marks sweating, salivation, another secretion, and small dose causes fall in the BP, muscarinic action. Higher dose may rise the BP. When it applied on the eye, it penetrates to the cornea and promptly causes meiosis by constricting the circular muscles, and when it constricts the ciliary muscles, it detaches the detach the contraction of the lens with the muscles and maybe decrease the intraocular tension, and it is commonly used by 0.5 to 4 up to the 4%, and it is also used by miotic, or you can say at the counteract of the mydriatics. Thank you, my dear friends. If you have any doubt, any queries, don't hesitate to write a mail, that is hnyadavyadav@gmail.com. I am further repeating my email ID, h and all are small, h and yadav@gmail.com. It is the autonomic nervous system is a very important chapter in pharmacology. Thank you very much.